Hello, I'm Jennifer Keller, the Programming Coordinator at the Westport Library. Today I'm speaking with Robin Wolfe Scheffler, who will share with us the search for infectious agents possibly responsible for cancer from the 19th centuries he writes about in his book, A Contagious Cause. This is the first book to trace the hunt for human cancer virus in America and links laboratory and legislature, creating a new chapter, Histories of Science and American Politics. Robin Scheffler is an associate professor in the program on Science, Technology, and Society at MIT. He's been a visiting scholar at the National Institutes of Health Office of History and the American Academy of Arts and Sciences. Welcome, Robin.
Thank you, Jennifer, for that introduction. And I really wish that I had the ability to be with all of you in the Westport Library to discuss my new book, A Contagious Cause. But I'm also very pleased to have the opportunity to talk with you virtually about some of the discoveries that I made in the course of writing the book and what they mean to me about how we understand the role of medicine in improving human welfare more broadly. And to set the stage for why the hunt for cancer viruses matter, I'd like to take you to a very different outbreak than the one that we have been accustomed to think and talk about in the last few weeks. And that was an outbreak that emerged in the Chicago suburb of Niles, Illinois. I'll just bring this up to full size. In the summer of 1963, newspapers carried accounts of a leukemia cluster in Niles, where the rate of leukemia was 8 times the national average. An official dispatched by the National Institutes of Health to investigate the cluster attributed it to an unidentified infectious agent. Another doctor was far more direct with the Saturday Evening Post, saying that leukemia looks to me like an infectious disease, a virus. And indeed, the thing that made the episode in Niles so concerning was not its uniqueness, but actually its ubiquity. The belief that cancer viruses and cancer clusters were caused by infection was throughout the United States in the 1960s. Indeed, Mrs. Ann Yockey of Westport wrote to a Nobel Prize-winning medical virologist asking if there was a chance that she could catch a cancer germ from her house after reading of similar stories in Life magazine. And this was more than a matter of curiosity or interest. It was a matter of great fear. If you look at this image, on your right, it's a young woman who's a survivor of the Niles outbreak, being visited by a doctor from the National Cancer Institute in 1966. And she was so concerned as to being stigmatized as a infectious cancer carrier that she refused to be photographed with her face towards the camera, to remain anonymous. But what coexisted with this intense fear of cancer as an infectious disease was also the optimism of what could happen if it was possible to develop a vaccine. And this indeed was the strategy that had worked so well against polio and soon measles and many other diseases. And it was in that spirit that the National Cancer Institute unveiled its, sorry, can you pause for a second? This super plan that the National Cancer Institute developed, the Special Virus Leukemia Program, drew on the most sophisticated methods available from military research and development to find a leukemia virus and develop a vaccine, making the development of this vaccine not a question of if Life magazine gushed, but when. And indeed, this ambitious program ended up absorbing more federal funding than was later dedicated to the Human Genome Project. And what I found truly remarkable as I read about this program in my research is that it operates for almost a generation without knowing that a human cancer virus actually exists. And it was episodes like these that I found when I was writing my book, A Contagious Cause, The American Hunt for Cancer Viruses and the Rise in Molecular Medicine, that made me think that this was a book worth writing and that there was something to be found by following cancer viruses through history. However, the deeper I delved into this topic, I realized that these cancer viruses were even more interesting than I had imagined. It wasn't just a story of viruses and vaccines. It's a story of how molecular biology migrates from simple bacteria to complex human cells, and with it, the promise of molecular medicine. And it's a story of how people debate and argue over what role government should play in advancing the welfare of society through medical research. And ultimately, it's the question that I ended up asking is how we understand the nature of success and failure in thinking about these programs and thinking about scientific research as a whole. And I'll come back to that at the end of my talk. But for now, for the rest of my time, I'd like to virtually take you through a few episodes from my book that help speak to these questions of what we understand cancer and contagion to be, and the efforts to identify the virus and their scientific and political legacy. So, we may be surprised to know that until very recently, cancer was not an especially well-studied disease. That changes in the early 20th century during something that historians of public health may have optimistically called the epidemiological transition. As you can see here, in 1900, the major causes of death were pneumonia, tuberculosis, gastrointestinal infections, all of which, of course, are caused by infectious agents. By the early 21st century, as we know all too well, the major causes of death are cancer and heart disease, which fall into the category of chronic illness. So there's one story here, which is the irony of bringing infectious diseases under control ends up making these chronic diseases more visible and more alarming for public health. However, within the toll of chronic diseases, cancer had always occupied an especially feared place. And that's not just because in the early 20th century, a diagnosis of cancer was likely to be a herald of your impending death. Tuberculosis, the leading killer of the 19th century, also carried with it the guarantee of death, but it was seen as a good death, a peaceful death, a death of gradually wasting away, whereas cancer has always been associated with nasty, painful, lonely death. So cancer was not just feared for the fact that it promised death, but also that it promised a bad death. Moreover, unlike the optimist interpretation that the control of infectious diseases had simply made society more aware of cancer, many other statisticians working for life insurance companies argued that actually rates of cancer were on the rise in the most modern industrialized nations, and the same nations that had managed to tame infectious disease were somehow engaged in activities, potentially pollution, that was causing more cancer. And this set off a wide-ranging and fierce debate regarding what the causes of cancer really were in an effort to understand them experimentally. Within this set of discussions, many were also eager and ready to apply the lessons of germ theory and microbiology to the study of cancer, and the idea that cancer might be contagious fit into a much longer running set of beliefs about the contagiousness of cancer. This is the blueprint of the New York Cancer Hospital. You can see this round ward here, which is actually a feature incorporated from infectious disease wards to slow the spread of diseases such as tuberculosis. And so the fact that it was included in the plan of this hospital, which becomes one of the seed institutions of the Memorial Sloan Kettering Cancer Center, is an indication that many were open to the idea that cancer might be infectious. Cancer's association with odor also buttressed this connection. One British doctor warned that the odor of cancer might spread through a house and bring the disease with it, and the best way of warding it off was with a stiff tot of brandy. And well into the 20th century, the idea that cancer might be spread by bacteria, insects, continued to have wide cultural resonance. The one place that the idea that cancer was contagious didn't really enjoy much support was in medical and scientific circles. And indeed, as you can see from this brief pamphlet excerpt from the American Society for the Control of Cancer, the idea that cancer was contagious was singled out as an especially pernicious and harmful belief to have about the disease. And the reason that the American Society for the Control of Cancer and other doctors thought that this was an exceptionally harmful belief was that if you believed somebody with cancer was contagious, you'd be even more likely to shun them and stay away from them. They'd be more likely to conceal the disease, and that in turn harmed the ability of doctors to detect the disease early. The idea of identifying cancer germs and promising quick cures was also very heavily associated with what the society considered to be quack medical cures. So indeed, in the early literature on cancer produced for public education purposes, they always start by reminding people that cancer is not contagious. But of course, the fact that they feel compelled to make that reminder is a sign of how widespread that belief was in society as a whole. all of this, of course, might have been resolved had science been able to produce proof or disproof that a cancer germ existed. But the activities of Peyton Rouse, a pathologist who identifies the first tumor virus in 1911, illustrates the difficulties of studying viruses in the early 20th century. Here's a picture of Rouse with an optical microscope, and I always think there's an irony to that because, as some of you might know, viruses are too small to be seen with an optical microscope, so whatever he's looking at in that microscope, it's not a virus. Rouse found that there was some agent he was able to isolate from the tumor of a Plymouth rock hen, and he was able to use an extract of that tumor to transmit tumors from one unfortunate generation of chickens to another. But despite the fact that he claimed to have isolated an agent, Rouse could not say what that agent was. He used a series of household water filters to pass the extract, and all he could say is that this agent, whatever it was so small, none of these filters would stop it. So he really couldn't point to a particular germ that could be identified, which made the microbiologists unhappy. Meanwhile, when he presented his results to cancer specialists, they said quite correctly that they didn't catch cancer from their patients. And so, frustrated by these experimental hangups and the skepticism from medical authorities, Rouse sort of gives up on this research in 1916. He's recognized 50 years after that in 1966 with the Nobel Prize for having discovered the first tumor virus. And that really reflects a lot of technical and scientific changes regarding the field of virology in between. Some of you may be familiar, for example, with the invention of the electron microscope, and you'd think, well, if surely the minute we can see a virus, that would resolve this debate. And that was exactly what a number of scientists in the 1950s thought, especially this Polish refugee scientist, Leon Domachowski, who claimed to have found images of viral particles in sections of tissue from breast cancer patients. And if you can't quite see where the virus particle is in that image on the left, on my right, you might be able to see it now that I put a red circle around it. But this illustrates actually one of the central issues facing people studying cancer virology in the middle of the 20th century, which is that new technology alone wasn't enough to establish consensus regarding the presence or absence of viruses associated with tumors. These images required a great deal of interpretation. There was the concern that there were artifacts produced by taking these electron micrographs. One virologist pointedly reminded Domachowski at a conference that images are unlabeled under the electron microscope. So simply following the science of virology didn't really provide me with a lot of understanding regarding why in the 1960s cancer virus theories and cancer vaccination experiences such a remarkable renaissance. And that's a story that takes me away from the laboratory and back towards the legislature. I should note that one of the other reasons that you wouldn't see a lot of talk about an ambitious government program to develop a cancer vaccine has to do with gentlemen such as these. These are members of the first National Advisory Cancer Council formed to advise the government's National Cancer Institute in 1937. And this was a very different National Cancer Institute than the research behemoth that you may be familiar with. This was a small organization, much smaller than any private philanthropic organization. And these were men, the elite of the American medical and scientific community, who were intensely skeptical of the idea that government could offer anything to science. In their minds, there were very few first-class scientists, and all that could be accomplished by pouring money into cancer research would be to conduct, in their minds, second-rate or bad science. And that's why, despite widespread popular support in 1946, for example, for a so-called Manhattan Project to cure cancer, the Surgeon General and the director of the National Cancer Institute speak out against this program. So even if there had been proof that there was a target of a cancer vaccination campaign, there's really no agreement within the medical community that the government should take a leading role in promoting that research. And what really changes is not the attitude of these men, but it's the entry of a new class of activists and advocates who become interested in biomedical research after the Second World War. And it was at this point in my work that I met Mary Lasker, who's really an extraordinary woman She may be familiar, once again, to some of you as the sponsor of the Lasker Awards, which today still recognize major breakthroughs in biomedical research. But she had a really amazing career before then. She was born in Wisconsin. She came out to study at Radcliffe and then spent some time at Oxford. She settled in New York, where she worked in both fabric design, art galleries, and public relations. And through that, she met her husband, Albert Lasker, who is an advertising executive best known for working on the Lucky Strike cigarette account. And Mary Lasker and Albert Lasker shared an interest in medical research. But Mary Lasker, unlike Albert Lasker, was also a committed New Dealer. And so her causes in the 1930s and early 1940s were things like access to contraception, and most significantly, national health insurance. And for those of you familiar with the history of health insurance in the United States, you'll know that the big push after the Second World War to establish a national health plan runs onto the shoals of accusations that it's socialized medicine. It runs into the opposition of the American Medical Association. And Lasker, after vigorously lobbying for this bill alongside many others, feels very frustrated. And of course, at this point, she could have simply decided that the effort had failed and gone away. She could have continued to push for national health insurance. But what I find so interesting about what she did is she shifts her attention to another area, another field of activity where the government could have a major impact on the health of the American people. And that, of course, is research into health, as shown by this wonderful cartoon. And for Lasker, the idea of biomedical research into disease seemed to be a more direct and easier way of benefiting the American people. She didn't have to go through the opposition that doctors expressed to a large-scale federal program. And the amount of money that she would be asking Congress to spend was very modest compared to healthcare as a whole. She used to say in her lobbying campaigns, if you think research is expensive, try disease. And finally, it was an area of appropriations that was still very much sheathed in the obscure rules of how Congress operates. And so through forming a partnership with one or two influential legislatures, Lasker is able to energetically help boost the budget of the National Cancer Institute. It quintuples between 1955 and 1965. And very soon, Lasker's power to lobby and can persuade Congress to spend money begins to make her and the sort of channels of public enthusiasm for cancer research the equal of the medical or scientific opinion that had previously directed research at the National Cancer Institute. And one of the prime examples of this is the drive that Lasker helps promote to do research into chemotherapy compounds. Chemotherapy was an area where an industrial approach to cancer research seemed to be necessary. All of these compounds needed to be screened as potential anti-cancer agents. And many scientists didn't want to do this work. The National Cancer Institute was unwilling originally to take a leading role, but Mary Lasker sort of foisted the program upon them, and very soon the National Cancer Institute became the single largest sponsor of chemotherapy research anywhere in the world. And that is sort of, a story of, growing government success. But once again, that success brings with it new questions and new challenges. In particular, scientists and biomedical researchers are now accountable to the American taxpayer to demonstrate that their research actually helps improve public welfare. And this idea is grasped upon by people who are small government anti-tax advocates, such as Democratic Representative Lawrence Fountain. And what Fountain does is he sort of realizes that he can't challenge the idea of doing research to make people healthy, but he can challenge the efficiency of that research. And the National Institutes of Health and the National Cancer Institute do him a favor in that sense, because the main mechanism through which they're dispensing all of this new money that's flowing in is something called the research grant. And the research grant is a very, it's ubiquitous today. People still get grants, but it's an instrument that's designed not to make sure a particular task is accomplished. It's an instrument designed to guarantee scientific freedom. And scientific freedom is literally written into these grants, which is to say that the scientists receiving the grant money don't actually have to do the research they're being paid to do. So Fountain hones in on this. He also finds many instances of misadministration. Stanford University uses a grant intended for research to build a pool, as only Stanford can. And so this idea that the money that's supposed to make people, the American taxpayer, healthy is being misadministered seems to be a really potent line of attack and causes a lot of concern among advocates of the National Cancer Institute and the National Institutes of Health. And this concern provides an opportunity for the then associate director of the National Cancer Institute, Carl Baker. Carl Baker gets into a career in research administration after he has such severe asthma that he can't work with laboratory animals as a biochemist. And he's very drawn to the idea of using a whole new set of management methods drawn from something called systems analysis, which is something that the defense think tank RAND comes up with. He thinks that he can use these methods to direct and craft cancer research to be accountable in the way that Fountain wants, while also providing justification for why the National Cancer Institute should continue to lead this field. And Baker decides to focus on cancer viruses as the first place to demonstrate the power of this systems analysis approach that he wants to take. And since Peyton Rouse had been doing his work in the 19-teens, a lot of stuff had happened with virology. The most important thing that had happened, though, was the success of the polio vaccine in 1955, and the massive campaign, the March of Dimes, had launched to develop a vaccine. Not only does this result in the successful production of a vaccine, it also vastly expands the capacity of virology to study many different types of disease. And indeed, many of the virologists who had successfully worked on the polio vaccine are beginning to look into the possibility of vaccinating against cancer and leukemia in the early 1960s. And these are the people that Baker turns to as he designs his program. However, Baker still faces the same problem Rouse did, which is that there is no human cancer virus that's been identified in a laboratory. And Baker's response to that is really to double down on these new management methods that he wants to use that are taken from the military. You can see here, this is the logo for the Special Virus Leukemia Program. I want to draw your attention to this dotted line between what are supposed to be viruses and what's supposed to be humans. These solid lines indicate causal connections between radiation and human cancer, between chemicals and human cancer that are already known. The dotted line, though, is evidence of a discovery that is assumed hasn't yet taken place, but it will. And the question, therefore, is not if it's worthwhile to build a massive program devoted to a cancer vaccine, but simply how quickly that program can work. As Baker's boss, the director of the National Cancer Institute, explains to Congress, it's not a question of if a human cancer virus exists, but when it will be discovered and how quickly a vaccine can be produced afterwards. And by reframing this as a question of management and organization, Baker and the National Cancer Institute are able to start spending millions of dollars on a cancer vaccination program, despite not yet actually having a human cancer virus to vaccinate against. And this money and these management methods are more than sort of just publicity. They end up building very real infrastructure to do virology and study cancer. This, for example, is Building 41 on the National Institutes of Health campus, completed in 1971. This is a building that cost many millions of dollars to build. It contains these sophisticated biohazard containment systems on the inside for working with human cancer viruses. But once again, no human cancer virus was not yet known to exist. So this is an example of the power that Baker's way of talking about management has for mobilizing resources and building infrastructure to study cancer viruses. And of course, we're all familiar with people who promise too much in an effort to get money. And that's why when I was sort of tracing the history of this program, I wasn't yet especially intrigued by this first observation. I became really interested when I saw what happened next. And that is what the program does when it doesn't find a human cancer virus. Because after 5 years of diligent searching, they haven't really made much progress. And what happens is one of the lead researchers is hunting for a human cancer virus for the program, Robert J. Huebner, who's a very well respected medical virologist. You can once again see the optical microscope, partners with another colleague, George Tadero, to make this really audacious proposal to account for the fact that they haven't found any viruses present in human cancer tissue. What Tadero proposes is that there's this one kind of virus, a retrovirus, which is an RNA-based tumor virus, which has these special components called oncogenes. And these oncogenes end up being inserted into normal cells during infection, and they stay dormant through generations before causing cancer. And that's unlike normal viral infection, where the viral particles can be immediately observed as you get sick. So this oncogene here is responsible for a cell becoming cancerous, but if you're using traditional methods for immunology or virology, you're not going to see viral infections here. And this idea was very controversial when Huebner proposed it in 1969. It might, of course, seem like special pleading. It provides a convenient explanation why, it's a theory that doesn't seem like it can be disproven or proven. It also, at the time, seemed to go against the central dogma of molecular biology in his proposal that RNA could edit the DNA of cells. However, in 1970, an enzyme is found which allows retroviruses to edit the DNA of cells. And that type of bold gesture that Hübner makes theoretically, which is proposing a whole new molecular mechanism to account for what he feels must be an administrative inevitability, and then having that hypothesis confirmed by laboratory science, is what's very much on the mind of many as they begin to contemplate the War on Cancer of 1971. The War on Cancer of 1971 quadruples the budget of the National Cancer Institute, and it also promises to take these methods of management that had only been used to study cancer viruses and make them the default way of combating cancer throughout the nation and across many different types of science. And this was an idea that was profoundly disturbing to the leading molecular biologists of the day, such as James Watson, the co-discoverer of DNA, and Norton Zinder, another widely respected geneticist. And this is because in the eyes of these molecular biologists who are poised between bacteria and complex cells, they're doing creative, unconstrained work. The idea that they would be told what to research, operating on these very strictly regimented government contracts that Baker used to direct research, was anathema to their conception of themselves as creative scientists. At the same time, however, because of the oncogene theory, these were exactly the scientists that the Virus Cancer Program and the National Cancer Institute wanted to recruit to study oncogenes and to study retroviruses. So it's really the ground is set for a major clash, which is why throughout the 70s, the Virus Cancer Program becomes a touchstone for debates over how much the government should manage biological research, and indeed whether or not biologists doing research should be expected to give clinical benefits or demonstrate the therapeutic benefit of their work. And this criticism and critique generally amounts to the idea that there's a field known as basic science, which deals with complex puzzles such as cancer. And this is something that the government can't intervene in because it's sort of too complex to manage. And also it may or may not actually have therapeutic benefits, and expecting to therapeutic benefits from that work is sort of an unrealistic idea. And it can only lead to, once again, second-rate science. So this is very much a site where molecular biology tries to defend itself from both medicine and from the government. The irony of this huge debate is that as it rages, two scientists at the University of California, San Francisco, J. Michael Bishop and Harold Varmus, are supported by the National Cancer Institute, and they end up discovering something that upends the entire premise of the program. They had gone looking for retroviral oncogenes, but instead what they find is this cartoon hopefully will help me make the point, is that rather than a cell catching an oncogene from a retrovirus, at some point what had happened is a retrovirus had entered a cell and caught an oncogene from a cell. So that means that the source of the oncogenes was not viruses, but an event that happened in the cell, and this had the effect of switching the orientation of the molecular study of cancer from searching for enemies without viruses to searching for enemies within, seeing cancer as something which is in our DNA, in a cell, and is best understood at that level, which also means that the study of cancer and the study of cellular development at the molecular level become cojoined. And this field blossoms rapidly in the 1980s on the basis of all the retroviruses that the National Cancer Institute had helped sponsor. And you can see the cartoons of these other scientists and their animal retroviruses down here, mice and monkeys and even a cat. And this is really what brings forth this powerful new framing of cancer as a genetic molecular disease and grounds so much optimism in the 1980s and 1990s, and indeed to the present day, that by unlocking the molecular keys behind cancer, that's the way that we're going to find the most effective cures for dealing with the disease. So in that sense, this is a story of serendipity. No one went looking for this when they were looking for a cancer virus, but they found it nonetheless. And that brings me back to these larger questions of how we understand success and failure in a case like this. And that really also depends on sort of where you stand, and I'll give you a few examples to conclude. So one irony is, I've said that they didn't find a human cancer virus, but as these events were happening in the late 70s and 80s, We, as a world population, were involved in the discovery of the hepatitis B virus and its association with liver cancer. And indeed, today there's a vaccine against hepatitis B that can help prevent many cases of liver cancer, but that's not seen as a success in the United States in the 1980s because, as this map suggests, there aren't that many cases of hepatitis B infection in the United States. It's a problem in sub-Saharan Africa and East Asia. So this is the question of who you are or where you are when you ask if something has succeeded or failed. And this is also equally true of debates surrounding how effective the human papilloma virus vaccine is in preventing cancer deaths. Another way of thinking about this is that the infrastructure of a program doesn't go away. In the 1980s, of course, our American attention is seized by another retrovirus, which is HIV and its connection to AIDS. And once again, many of the researchers in retrovirology who provide some of the foundational discoveries for dealing and confronting the HIV/AIDS epidemic were trained by the war on cancer. And had they not been there, our response to AIDS would have been much slower. So this is not a case of failure, but these institutions and this infrastructure that we build for studying one type of disease really can always carry over and help us with another disease. A final way of thinking about this has to do with the political work that talking about failure does. And as the Human Genome Project is being discussed, or even later discussions of large-scale biomedical science, the virus cancer program is used as what becomes a touchstone memory of failure to cut against the idea that government should ever take a role in managing biomedical research, and that really should be left to these new entrepreneurial biotechnology companies. And throughout this, I think these are all important ways of understanding how ideas of success or failure work and how we relate as citizens to biomedical research, but it's also what we even lose by thinking about biomedical research in the laboratory. As this cartoon, of course, aptly illustrates, when we look in the laboratory, we're looking away from a lot of the other major causes of cancer in society. So this is from the 1970s, and you can see once again a researcher with his microscope being asked to do something for tobacco and polluting industries. And this really speaks to the challenge of how we even frame disease or use science as a lens for dealing with what is really a much more complicated social problem. And that unfortunately comes back to the fact that one of the biggest factors in how long you'll live with or survive cancer still has to do more with your zip code and where you are in the United States or in the world than with the biological nature of the disease itself. So all of these questions of success or failure in the laboratory touch on this broader issue. And that's where I'm going to leave you this presentation. And with A Contagious Cause, it's not just a story about our success or failure against disease in the laboratory, it's about how we as citizens understand and use science in conjunction with many other forces in society to make ourselves as healthy and well as possible. Thank you very much.
Thank you, Robin. That was very eye-opening and I learned a lot. So thank you very much. For those of you watching at home, remember that you can buy A Contagious Cause from your favorite retailer right now. And for other author talks and educational programming like this, please visit westportlibrary.org. Thank you so much, Robin.
Thank you.